Recording.
All right, everybody, welcome back here. Spectre here with another edition of the Think Tank here. Continuing on from the first, we still have Soko, aka Laura, in the house. We got Homesteader, Drunk Pinata, and of course TMC, our resident expert on genetics and the genome and the manipulation thereof. And we're going to discuss a little bit about that here tonight in conjunction with some stuff on chimeras. So, TMC, if you've got something ready, take it away.
Okay, so there's a whole lot of information to go over. So we're just gonna kind of pick away at it and see what piques the group's interest here. And we're gonna start with this thread that I put together long time ago, and it's on George Church. George Church is from Harvard. He's attributed with mapping the human genome, world renowned. He's basically publicly touted as like the best geneticist around. So there he is. Look at his face. This guy's an interesting fellow. So what I want to start with, since we're going to kind of deal with chimeras, we need to understand, I guess, what genetic engineering means to us as people, as humans. And really where we're going to go over the next short time is just that. What can be done to humans that alters and modifies your genetics, your DNA? And this loosely ties in with the whole mRNA and other type of RNA style vaccines that are failing. They're failing forward. No pun intended.
At TMC,
Yeah.
I guess before we get too far into this, maybe for the audience and that listening, if they're not as clued in, can you briefly explain the difference between just plain old genetics versus another common term we hear like epigenetics?
Yeah. So your genetics would be basically the building blocks or the software code in your cells that tell the rest of your body what to do. And it's all the software is there, it's in your DNA. The epigenetics piece is whether or not specific genes are turned on or turned off. And epigenetics can be manipulated by your environment. They can be manipulated by stress, by food. They can be manipulated with electromagnetic energy. They can be manipulated when you go through rites of passage. Toxins can manipulate them. So they do things like change the way proteins are expressed in your cells, for example, amongst many other things. So the epigenetics piece is just as important as the genetics piece, but for different reasons. And that's really the rub that I see. And I'm by no means a genetic expert. I've spent a lot of time reading about this stuff, and that's really all I can tell you is that I've spent a long time reading about these kinds of topics, trying to figure out how they want to wire us up to the internet with nanotech or synthetic biology, biotechnology. And it's going to be a combination of both of those things in my view. But the real rub, I think, why these vaccines are having problems is because they don't really understand the epigenetics piece. They can, they can cut and paste genes, take some out, put some in, no problem. But what it does epigenetically, how they express themselves in your body, that's why people are, dying from these trials. To give you an idea of where this is all going, genetic manipulation, um, there's things like DNA computing and a whole bunch of other things that are in this thread on Human Internet One. If you go to Twitter, they can encode information in DNA, use it as storage drives. And what I want to show you is, well, here's an IARPA program that discusses mirroring or matching the um, learning, if you will, by I guess pairing up AI comparing and contrasting AI neural networks with the brain's neural networks. And genetics plays a huge role in that. What I want to get to is this right here: mapping neural connections, genetically engineering mice so that each neuron is barcoded throughout its entire structure with a unique RNA sequence. And this is a Weiss Harvard Institute document that discusses, this is IARPA, see? IARPA funded brain mapping consortium, 21 million from IARPA to discover the brain's learning rules as synaptic circuit design. So this idea of mapping the brain and barcoding the brain so they understand how it works so that they can make AI function like the brain. And the real takeaway from this is this idea that George Church, right, professor of genetics at Harvard and professor of health sciences and technology at Harvard and MIT, right, to map neural connections, the consortium will genetically engineer mice so that each neuron is barcoded throughout its entire structure with a unique RNA sequence. The technique called bionic barcoding of individual neuronal connections, developed by Anthony Zador at Cold Springs Harbor Laboratory. Thus, a complete map representing the precise location, shape, and connections of all neurons can be generated. Now, this is kind of foundational before we get into why, and
I
when we get
guess
into yeah.
I guess before you get into that, TMC, can I pose a question?
Mm-hmm.
In your general estimation, how far down the path of genetic manipulation would you say that humankind is at this very point?
Pretty far.
And the reason I ask that is this, and this is going to be controversial. From the standpoint of what we're seeing play out in the populace of gender manipulation, things of that nature, where you have manly women and womanly men, would one surmise that there has to be something going on along the lines of genetic manipulation for that to occur?
That or epigenetic manipulation, yeah. And that's it's really it's really the crux of it all, right? How far down the rabbit hole are we? Who knows? I think we're pretty far down the way, and it's
I
done
would
with
agree.
chemicals to it's done with chemicals to interfere with your genetics or epigenetics, and it's done with the food. It's done with what they put out into the atmosphere, and then it's also done with vaccines and lotions and shampoos and deodorants and
Makeup.
everything else. Yeah,
Yeah,
it's and
I
they're
don't mean to come off funny about this, but Alex Jones was actually right 4+ years ago when he was talking about turning the frickin' frogs gay. And they actually did the little cutout piece where they showed that atrazine runoff from the fields got into the water, and the water consumed by the frogs actually did start to swing their door the other way.
yeah, and those are basic forms of manipulation. We're going into this stage where what you're seeing on the screen, for example, DNA encapsulated payload can be delivered and specifically target cell types. Could be a heart cell, liver cell, kidney cell, could be a brain cell. It could be a specific location in the nervous system. And they bond to that location and they open up their genetic payload and they open up the vessel for the genetic payload and then they deliver whatever's inside. And typically it's either medication or specific genetics. And this is nanorobotics, nanomachinery. When you think of a nanorobot, it's not some mechanized thing that's got scissors and stuff on it. It doesn't work that way. You're dealing with enzymes and proteins and chemical reactions and acid changes and all these things that are done at in the biological sphere. And they're machines in the sense that they do something that is functional, that is intentional, that is designed with an intended outcome. And
and
this
predictable,
is
right? Like that's there is an intended
right. intentional. Whether or not it's the predictable part is, I think, failing, which is what we're seeing with the whole vaccine trial thing at this point. They can model it and have predictions based on their modeling, and they have very sophisticated modeling technology for this stuff. But as it actually, as it actually works, we're seeing not so good outcomes at this point with the types of cocktails, so to speak, that they're putting together for this quote unquote pandemic. And it's really sad because they're forcing us into this position where most people are going to want to feed their families, so they're just going to do what needs to be done. And the more damage that occurs, the more people are going to resist, or the more they're going to buy in to this premise that they made the right choice. And if it damages their family, they're just going to use that as motivation, so to speak, to continue through this evolution. And you have this CRISPR, which is how they get in. CRISPR-Cas9 is just one method of CRISPR that they use CRISPR technology to go in and unzip your DNA and add new DNA, remove DNA. And I mean, this is all Harvard-based stuff, but this is their gene drives. And with gene drives, they can do things like start editing your germline, meaning they edit your offspring. No joke. And I'm gonna stop share and make sure that I've got my sound enabled, and I don't. I want to share this with you because these are vectors by which this stuff can be administered, and it can be administered. It can be administered through open air delivery, or it can be administered through things like vaccines, potentially through food. But this is kind of where they're looking, and in my view, they're already here, and it kind of speaks for itself.
Historically, scientists have been able to alter the traits of domesticated plants and animals, but have not been able to do this to wild populations. Here we use mosquitoes as an example to explain why most genome alterations designed by humans don't persist in nature, and how a recently proposed technology could change that. The transgenic mosquito, tinted blue, has an altered gene inserted into one of its chromosomes. When it mates with the wild-type mosquito, each parent contributes one copy of each chromosome to their offspring. Thus, only 50% of offspring will carry the altered gene, while the other half will inherit the wild-type version from both parents. Even if the altered gene doesn't reduce the likelihood of each mosquito surviving and reproducing, it may persist at a low frequency in the ocean of wild mosquitoes, or it might go extinct after several generations of especially unlucky inheritance. This process is what keeps us from altering wild mosquitoes to prevent them from transmitting diseases such as malaria or dengue. A team led by Kevin Esvelt at the Wyss Institute, the Harvard Medical School, and the Harvard School of Public Health has now outlined a way to build gene drives that can improve the odds that almost any altered gene will be inherited, potentially allowing them to spread through even wild populations. The proposal relies on the CRISPR-Cas9 system, a new genome editing technology co-developed by the same researchers at the Wyss. Gene drive mosquitoes carry both the altered gene, the genes for the Cas9 enzyme, and several guide RNAs that tell it where to cut. When passed to offspring, the guide RNAs direct Cas9 to cut the wild-type version of the gene inherited from the wild-type parent. The cell then copies both the altered gene and the drive when it repairs the damage. Because the mosquito now has two identical copies, one on each chromosome, all of its offspring will inherit the alteration in the gene drive. This same process will be repeated in subsequent generations, causing the alteration in the gene drive to spread through the population.
Yeah, so what you just saw there is open air genetic manipulation of an entire species. I mean, they use mosquitoes as an example, but what does a synthetically designed biological outbreak look like? And then the question becomes why? And what happens And every time you get sick, your body builds an immunity and you get coded with some type of new software, some type of new RNA or DNA software. And this is the new world that we're living in. This idea of subtle, soft, uh, um, biological warfare, basically. That's the world that we're living in. And
So the response in that code is that it like that code is would be within me, within you, if we, you know, if you're exposed to something and it had a response, is that code the same code amongst everybody?
yes and
I mean,
no. Go ahead.
I suppose, I mean, like, and I think we you and I have maybe have touched on this before, where you're basically might be imposing some sort of commonality or some sort of common code between, inserting it into every person so that it could be manipulated later.
Yeah, we kind of touched on this idea of a multi-part system that will assimilate over time. And yeah, that's what I really think's kind of happening. So there's some links that if you go to patriotsoapbox.com, these are posted at patriotsoapbox.com underneath the shows tab. You can go look at all these links yourself. I'm not, it's free. All this stuff that I've put together is open source information. Um, so maybe I should show you where that is for now, so that if you choose to go kind of explore this world of genetics, um, you can go here. So if you just go to patriotsoapbox.com and then you go over to the shows tab up here, menu, or the three horizontal lines if you're on a mobile device, um, you go down to shows and find the show banner Big Tech, and it looks like this. And if you click on this link, you will see a section that says read, watch, and think for yourself. If you're on a mobile device, you're gonna have to scroll down. It's gonna be towards the middle or the bottom of the scroll. But if you're on a computer, it's off to the right, and you click the notes, and it takes you to a G drive where all the shows are broken up in increments of 10. Start at 41 and look at 41 through 50, 51 through 60, and 61 through 70, and you will see nanotechnology and biotechnology, um, genetic manipulation. All of those things are here. And there is a lot of information to go through. Um, Interesting. That's why. Okay. So what you see here is just link after link after link about Pentagon and high profile institution, this, that, and the other. And it's really too much to kind of get into in an hour session. But it's this is the whole foundational piece, I guess, for chimeras and genetic manipulation and what I think they want to do with people. For example, the US military agency invests $100 million in genetic extinction technologies from The Guardian. Just, we're not going to read it, but I'll show you that this US military agency invests $100 million in genetic extinction technology. And they sell it. Technology could be used to wipe out malaria carrying mosquitoes or to other pests, but UN experts say fears over possible military uses and unintended consequences strengthen the case for a ban. Genetic extinction technologies.
That's basically manipulating a mosquito instead, but to drop like a payload, right? To give like an injection.
Yeah, so that's exactly right. If you take the idea of trying to genetically extinct a species, what you're talking about doing is introducing new software into a biological that then propagates throughout the entire species. What if the biological information wasn't intended for them? So you have a multi-stack approach. You deliver the information into the genetics of a mosquito, which then goes out and bites animals or humans. transfers that genetic information into another animal or into a human, where then that genetic information makes sense to that genome, makes sense to that species. So you could piggyback, for example, human genetics in mosquito genetics, whereby it is dormant in mosquitoes. This is a hypothetical example. It's dormant in mosquitoes. And then when it comes into contact with the human, bites a human, the genetic information is shared with the human. Now all of a sudden the human understands that genetic information that was dormant in the mosquito, and then it could potentially do something. Are we that sophisticated yet? Not really sure. I don't think we're quite
Well,
that far.
And think back before this particular, using air quotes behind the scenes, pandemic here, right? What were the previous two big health scares that they tried to whip up a frenzy over? Well, we had Ebola. And then around the time of the Olympics down in Brazil, we had the Zika virus. And the Zika, of course, as people may or may not know, I believe, was largely transmitted by mosquito. And of course, around that time frame when the Olympics were going on down there, one of the things that we heard people doing, and even here stateside in Florida, aside from spraying masses and that to prevent against Zika spread, was the fact that genetic mosquitoes and that were going to be put into the population.
Yeah.
Well,
It's Bill Gates has been working on using mosquitoes for vaccines for a while. I mean, he's funded that openly.
They've been using that concept of genetic extinction for the longest time on pests, longest time, and that's exactly they typically through the introducing a sterile male that basically has sex with the female of the species, but she cannot produce, and by design, it it cuts down the population. They do it with mosquitoes, they do it with flies, they do it with all kinds of different pests.
Well, anytime you have an intersection with the Clintons and Gates, well, you have to wonder.
Yeah,
because I think it was Chelsea Clinton was saying something about weaponized mosquitoes, right?
well, and I mean, in a way, you don't even have to go down that far. It's an aspect of the question is: do you want the mosquito to be the carrier or the transmitter? Because that's how mosquitoes deal with malaria. The mosquito is a carrier of malaria. They don't have malaria. They suck the blood out of a human that has malaria, give it to another human by stinging them, and transmits the malaria. Same concept could be done easily enough. Introduce an infected individual in a population, and you have a predisposed mosquito, excuse me, altered or otherwise, and there you go. It immediately.
Yeah, it's and that's the difference is really how sophisticated is the delivery.
Correct.
If it's through a vaccine, I think the sophistication can be better, or it can be more sophisticated because you have a very controlled environment. But with open air modifications like a pandemic or mosquitoes or whatever, I don't think you have those luxuries. So what you're just kind of scrolling through here, looking at this in real time. All this stuff about DNA, single strand, double strand DNA, making origami. This is, for example, if they were to introduce something into your body, this idea of DNA and RNA origami, where they can assemble this, insert this, assemble this, or tell your cells to grow something like this, and then they know what it's going to do. So they can take a DNA strand, and the DNA itself, with its instructions, will actually fold and shape into a unique scaffold or a unique shape or a unique whatever, and create what they call DNA origami. It's just a basic example of something that could potentially be self-assembled, self-assimilated in the body, And again, this is foundational for some of the articles that we're going to look at in a little bit when we start looking at chimeras, because the idea of an mRNA vaccine really begs the question, are they making us chimeras? Are we becoming chimeric as a result of genetic manipulation? And that's a real issue here, because you're kind of messing with God's creation here.
Yeah, this
DNA
is
nano.
nothing new. This has been happening since 1974. This is when the actual first that was first published about was in 1974, where they were already
Right.
able to create human fetal lungs, kidneys, pancreas, all in a nude mouse. So, I
Yeah,
mean, this has been going on for decades.
and I guess in conjunction with that, we've been tipped off in the past by
A
what
recent development
exactly
in nanotechnology
23andMe and
allows scientists to build
Ancestry.com are doing too.
Yeah, this genetic collection is huge. And I mean, I don't know if we're going to get that. I don't think we're going to get far enough to be able, I can show you patents where they literally can read and manipulate your genetics with electromagnetic energy. They just take the subharmonic of your genetics and whatever gene they want, they can identify it and then manipulate it using a subharmonic resonance of it. And so we're rapidly moving into an environment where we are the open air experiment, and the DNA collection, for example, the PCR test for testing for COVID, normalizing this idea of handing over your genetic information to be then cataloged. Can you imagine what will be learned when they start delivering vaccines and then mandating more tests, and or they have other types of flu and cold strains that get put out there that may or may not be publicly disclosed, and then we're just randomly, or we're not randomly, we're regimented into handing over our genetic information with these tests.
Well, I hate to
The
say it,
amount
but
of information. Let's go ahead.
I was going to say, I hate to say it, but where my mind immediately goes is what better way to get us down to a population of 500 million as advertised on the Georgia Guidestones.
It's that, and it's that, as well as going through this process, being able to understand what the other genetic characteristics are, how they're expressing in the body. To that point, we're going to move forward, and I'll
As
show you
far as
what
the
I'll show you what
harmonics
I
go,
mean.
because I want to the resonances, just before we get off that topic,
Mm-hmm.
are you because there's two ways you can go. You can with the harmonics, but also the fundamental frequencies. Considering that even like power lines can be influential, which would be well below the frequency range by many factors of what the actual resonance of the DNA would be. Um, is that what you were more referring to as far as the lower, ultra low frequency type stuff, or are you more referring to more ultra high frequency?
It's all of the above. It can be done with sound waves. It can be done with electromagnetic waves. I think this is it. Methods for determining therapeutic resonance. This is one of them. There's two of them that are really, really interesting patents. This might be it. This one talks about determining resonant frequencies that can be used in therapeutically or beneficially for debilitation of specific types of genomic materials, including DNA, RNA, genes, or gene sections. Can be used in a variety of circumstances, human and animal disease conditions, therapeutic resonant frequencies for use in various media having different refractivities. So let me see if this has subharmonics in it. Yeah, it does. This is the one I was thinking of. So they talk about using a frequency generating device that's cheap and available. Your phone is a frequency generating device. It generates sound frequencies. It generates electromagnetic wave frequencies. And what they're talking about in this patent, we're not going to really go through it, is this idea of dividing the resonant frequency of a gene, of a protein, of a specific section of DNA they've lined out how to measure its distance, its size, if you will, and then apply a frequency to it, and then divide it by 2 until you get into the range of what the transmitter that's nearby is able to emit, all the way down to, here's an example of 636 hertz, which is something that you would hear as an audible sound coming out of your phone. It can also, 636 hertz can also be emitted from the antennas that are in a phone, for example. I'm not saying this is happening. What I'm saying is this patent specifically discusses this ability to, with very basic math, taking a division of 2, so a certain resonant frequency divided by 2, divided by 2, divided by 2, right? Subharmonics, to get within a frequency band that is available with a frequency generating device nearby. And it's not as simple as I just explained, because as you do these divisions of 2 and you get into these subharmonics, you have resonance loss. You still get resonance, but you may have to increase the number of pulses, or you have to increase the power. There's a lot of other things that take place. But the point is this: the ability to resonate your genetics to do things, as they say here in this patent, to be able to do things that like provide therapeutically or beneficially relevant information or therapy, right, to your cells for debilitation of specific types of genomic materials, including DNA, RNA, genes, and gene sections. This is here, and it's real. And what I see from this, I'll read this piece and I'll tell you what I see. Therapeutic or beneficial resonance frequencies thus determined are adapted for use with currently available frequency emitting devices by shifting resonant frequencies to electromagnetic ranges capable of generation by such devices. So if our DNA is resonating in the infrared range, for example, a few divisions of 2 puts us down in the millimeter wave range. A few more divisions puts us down in the radio frequency range. A few more puts us down into the ELF range, and then down into the sound frequency ranges. And these are potential remote triggering devices. That's what I see. So we have this idea of being able to resonate your DNA, but we've got the genetic information that needs to be put in there in the first place. So this can manipulate the epigenetics piece you were talking about earlier, Spectre, and it also can manipulate your genetics. So it manipulates your epigenetics and your genetics. And there's another patent that's related to this. It's microwave resonant therapy or something like that, that talks about being able to do just that, be able to interact with your epigenetics.
Well, I wonder, with the different types of intensity, but depending on which frequency band you're in, if you're going to be in that lower harmonic, you're going to get a broader effect on maybe like a multitude of different types of genetic information. But if you're in a more high frequency range, it's much more directional. So it's going to be directed at a specific, a specific type of modality, right?
No.
Nope.
And I'll show you why. BrainQ Technologies, therapeutic electromagnetic fields. Let's see.
I mean, while you're looking for that, there's a reason I believe that J.P. Morgan and such stopped funding Nikola Tesla and made sure to break him.
So it says in this patent, I'm going to leave whole here highlighted. This patent right here is method and device for enhancing brain activity. And this talks about resonating specific neural circuits of the brain or specific centers of the brain in order to steer stem cells, brain stem cells, over to that area to then regrow damaged tissue, traumatic brain injury, a lesion, surgery, whatever, where you have a missing piece of brain or damaged piece of brain. And using electromagnetic fields, they can steer the new stem cells over to the area of interest, deposit them, and then stimulate neural circuits to then get the brain to rewire itself. And to your point about the having one frequency range versus another and having that be more sensitive or less sensitive, that's not true. It may impact frequency and or power requirements as two examples, not exclusive, but two examples, but it doesn't necessarily mean that it limits the specificity, like the accuracy, if you will. A scalpel instead of a sledgehammer, it doesn't matter. It can be a scalpel the whole time. It's just the mass of the scalpel might be 2 or 3 times more than another adjacent scalpel based on the frequency. If you're
Well,
closer
maybe
to
a better
resonance.
analogy I was trying to go for was more of like the because it was more of a bass harmonic, that it would be mildly affecting a broad range as opposed to specifically targeting a specific range.
I see what you're saying, and yes, it's possible, in my opinion, that if you go into the subharmonics, you may impact, you may resonate more than one specific component. It may not be as intense as one versus the other. There may be a dominant or a primary resonant component, but you could. But the reason I'm still going to say potentially that doesn't matter is really this. It kind of explains it out in this patent that discusses being able to enhance brain activity. The transmission of electromagnetic waves do not have to be targeted to a specific region of the brain. Instead, the transmission of a particular frequency over the whole brain will exert an effect on the relevant nervous system alone. Therefore, nervous system having different operating frequencies will not resonate and therefore will not be active in response to the transmission. So there's a whole lot more to consider than just frequency alone, I guess is what I'm getting at.
No, I get it. I think we're on almost on this, because this kind of, I think, clarifies the question that I was asking, because I was like, yes, you may have a broad range and you may have some, if you're talking about that bit, the lower base frequency that's more of trying to, that could stimulate other areas broadly, but there are certain ones that are definitely more susceptible to that lower energy, or just whatever energy you're putting into it. Okay, I lost my train of thought.
Yeah, no, you're right. You basically, that was a good place to stop, because that's exactly right. It may impact all, but if done properly, it's going to impact something specifically a whole lot more with a whole lot less output or a whole lot less energy than anything else. And that just, I think, is a representation of the sophistication and or refinement of whatever it is the technology is that's being used.
Yeah, I'm gonna have to look back on that, because I think I understand what you're saying. It's gonna take me a second to register.
Sure. Yeah, man, I totally get that. So let's move into genetic modification and some of the specific subsets of it. And then we're going to try to tie all these pieces together. This idea, we'll start with optogenetics, this idea of using light to interact with nerve cells, neurons, whether they be in your nervous system or in your brain. And this idea that they can go in and add genetic information into your cells to then have them express light sensitive proteins. And let me see if, I don't want to spend too much time on each one of these, but there's Ed Boyden he's an MIT professor. He's at the top of this thing. This is an 8-minute video. This might be too much. Selective brain stimulation with light. MIT.
My name is Ed Boyden. I'm an assistant professor at the MIT Media Lab and also in bioengineering and neuroscience. And I lead a group that works on methods for controlling brain computations by using pulses of light directed at individual cells or at sets of cells to figure out how they work. If you can control cells in the brain, you can figure out what their power is, what they can influence. And also, if you can control cells in the brain, you could potentially fix aberrant brain states. You could come up with new kinds of therapy, resculpting the computations that have gone awry in a neurological or psychiatric disorder. So starting back in 2000, I was very interested in collecting molecules that would allow us to control the brain with light. And there's a class of molecules that are naturally occurring that convert light into electrical energy. They're sort of a primitive form of photosynthesis. And of course, the brain is an electrical device. So if you can put these molecules that convert light into electricity into the brain, you can now control the brain with light. The brain's very heterogeneous. Unlike a computer, which is made of just a few circuit elements repeated over and over again, the brain is made of thousands of different kinds of cell. And these cells have different shapes, they have different molecules in them, and they project to different targets. So if we can target just one kind of cell with light, then we can turn it on or off, and all of its neighbors will be unaffected. So we can figure out just what that one cell type does, and if it's a cell type that's going wrong in a given disorder, we can fix it. We use reagents that come from nature. So there are many bacteria, archaeobacteria, funguses, plants, and so on. Pretty much every kingdom of life, except for animals, actually, has these molecules that sense light and can convert them into electrical signals. These molecules sit in the membranes of cells. You can think of them as like little solar panels that coat cells. And when light hits them, they translocate charge from one side to the other, and that, of course, is an electrical signal. So we find the genes for these molecules. These are all genetically encoded. In these organisms, the DNA contains the recipe for making these molecules. So what we do is we take the DNA that represents the code for how these proteins are made, and we can put that into a little virus or a little cassette. And then that cassette we can put in any cell. We can put it into an animal cell, we can put it into a human cell, a cell line. Whatever we put it into, that cell now becomes controllable with light. By doing this, you can think of it as that cell that we put this little cassette into is now installing those solar panels on its membrane. It now becomes sensitive to light and is controllable with light. So there's two reasons why we want to use this tool. We want to use this tool to understand how the brain works, parsing out how each part talks to the other. And also we want to prototype therapies. We want to figure out what are the targets that can be used to fix brain disorders. Now, of course, in the 20th century, there are many different technologies for addressing brain disorders. Pharmaceuticals, of course, which have been immensely impactful, although they often have side effects, and electrical stimulation. Electricity, of course, will go in all directions, right? It goes down the path of least resistance, which is partly where...
All right, so you get the idea. They find genetic information that transmutes light into electrical energy. They piggyback it on a virus. They deliver the virus into a host, and then that host then expresses the genetic information to allow light to be converted into electrical energy into the body to ultimately interact with things like your brain. So that's optogenetics in a nutshell. Let me see if I can find a cool picture that may help show. This is the Mcgovern Institute. Very interesting website. Light-sensitive proteins allow scientists to study multiple sets of neurons and how they interact with each other. Optogenetics is a technique that allows scientists to control neurons' electrical activity with light by engineering them to express light-sensitive proteins. This is the major way or platform by which they've been studying the brain. There are others, and we're going to go over those right now. So you've got optogenetics, the idea of manipulating brain cells with light. And there are different pieces to this puzzle. Since we're talking about light, you have these things called upconverted nanoparticles, quantum dots and upconverted nanoparticles. This stuff is real, man. So when you think about what they might put in something like a vaccine or something in a cosmetic, right? You can buy this stuff online. And an upconverted nanoparticle, what it does is when it is energized with sound or light or with electromagnetic energy, it emits a specific frequency of light, a specific type of photon. And you can buy them for whatever you want. Green quantum dots is in here. They have green, yellow. We've got other ones as well. and they have these glow dots. This is really interesting, this idea of glow dots. Because glow dots, based on what kind of energy you, here, I'll just say this, that you have different types of glow dots and they emit these particular wavelengths of light. So you can have nanoparticles in the body that float around or deposit, and when they're excited, they emit these particular frequencies of light sitting right next to potentially genetically modified brain cells that are designed to emit a voltage or go through an action potential, which basically says that your neuron is firing when they see a particular frequency of light. And you can see 1, 2, 3, 4, 5, 6. There's 6 different frequencies here, and they can encode for whatever they want if they have the right genetic information. And so the combination that I see is really a blend of genetic manipulation, but in addition to nanotechnology, synthetic nanotechnology, so that you get a very robust, complex bidirectional interface. There are other ways to do this. And again, if you're really interested in this, go look at this stuff yourself. There's pages and pages of notes here that they're link after link after link after link. Closed loop functional optogenetic stimulation. Sonogenetics, controlling memories with ultrasound, controlling brain cells with sound. So we talked about light. Now we're going to look at something that the study of manipulating cells to be more responsive to light, or excuse me, to sound, pardon me. And what they do is they're able to basically make cells more sensitive to sound. And they say that right here, we discovered that a pore forming subunit of mechanotransduction channel TRP4 increases the concentration of calcium ions within a target cell in response to ultrasound. Increasing calcium ions activates neurons and contracts muscles, perhaps even alters immune cells or other cell populations. So when they find these ion channels, which are the things on the outside of the cell that Ed Boyden was talking about, these ion channels come in different flavors. Some respond to light, some respond to sound, some respond to heat, some respond to pressure, and on and on and on. Well, they can target each one of those, and they can tell your genetics to grow more sensitive ion channels specific to a particular thing. Sensitive to light, sensitive to sound, sensitive to heat, sensitive to magnetic fields. And this is the subtle, gentle genetic manipulation that is possible in today's age. So let's see what this is. 3-minute video. Let's see what it says.
Sonogenetics is a new technology. This technology is used to manipulate neurons within the body, and we are using ultrasound as a non-invasive trigger to control neurons that are expressing proteins that we are delivering to them. One of the big applications that we think this could be useful for is to replace deep brain stimulation. So currently, deep brain stimulation is used to alleviate symptoms of Parkinson's disease in humans. What we think we could do is replace that entire surgical invasive procedure with something that's non-invasive. The way this technology is going to work is, let's imagine that you're trying to control a population of cells that are within the brain, and this population occurs deep within the brain. So what we would first do is deliver a protein to that population using a delivery system, often a virus, and that virus will initiate, infect, and deliver proteins to those target cells. And now these target cells are modified such that they are now sensitive to ultrasound stimulus that is coming into the body. The ultrasound stimulus is provided by a transducer that sticks outside of the body that can deliver the energy that is required, the ultrasound itself, through the bone and the skin to that target. So on demand, whenever you turn this transducer on, you would deliver a stimulus which would then turn on that neuron. At the moment we are activating neurons, but we are hoping that we would find a channel that will also allow us to turn neurons off. and that would then give you both activation and inhibition inside the brain.
Some of the diseases that sonogenetics could potentially be useful for would be the treatment of Parkinson's disease, neuropathic pain, post-traumatic stress disorder, and potentially movement disorders such as paralysis. This is a potentially really exciting technology development that will allow us both to answer new and important research questions, as well as potentially transition the technology to use in humans, which could radically improve how we treat some current neurological conditions that are difficult to address.
We're building novel, brand new custom-made instruments to help us interrogate this. We're collaborating with scientists outside of the Institute around the Mesa to really help us understand and use new tools to really interrogate the mechanisms, really understand the mechanisms on how ultrasound is interacting with the membrane and the proteins that we deliver to give us this control over the cells and tissue.
We coined the term Sonogenetics to describe this technology. We are excited with the results that we have obtained. We're excited with the prospects of this technology. We think that this is something that could revolutionize the field of both neuroscience and medicine.
So that's in San Diego, I believe. So that's being able to manipulate cells with sound.
That's why I brought up Tesla.
So here's an article, Sonogenetics in a rodent brain. They're far along with this stuff. This is far along. I'm trying to see a date on it. Posted January 28th, 2020. And let's see if it's got any cool pictures in here. 33 pages. So here's focused ultrasound applied to the brain. And what it does is, without ultrasound, you have this ion channel. That's the thing on this is your cell wall. So this neuron right here, this is the outside, the perimeter of it. And when they apply ultrasound to it, it opens this up and causes the neuron to fire. Or like they said, they want to be able to find ways to use ultrasound to turn brain cells off. So you have an on and an off switch, a 1 or a 0, an up or a down, a left or a right. And then when you map out where all the brain cells go, which ones talk to what and what centers are activated, multiple centers of the brain being activated at the same time to do things like produce complex emotions associated with visual imagery or memory or whatever, you can then begin to start delivering sophisticated stimulus to then have controlled active hallucinations that result in full sensation, full sensory experiences, where they can engage your sense of smell, your sense of touch, your sense of taste, what you see or visualize in your brain, what you potentially hear, as well as an emotional experience, all at the same time, to create a complete, fully immersive, active, controlled experience or hallucination with artificial stimulus. And you think, oh yeah, it's focused ultrasound, I just won't buy it. When you look at how electromagnetic waves, for example, interact with the body, you get things like thermoacoustics, where the electromagnetic energy, as it passes through your skin, skull, and tissue, it gently heats it and causes a mechanical wave or a pressure wave to be created. And if you pulse it with the right frequencies in the right way, you can generate focused ultrasound. You can generate ultrasound in the brain. And that's the real rub that I have with the electromagnetic grid that we're putting up. It does so many things. It can heat tissues up, and we'll get into thermogenetics here in a minute. But it also generates pressure waves. And you have the voice to skull technology that the Air Force has 2 patents for, as a matter of fact. Thermoacoustic hearing, basically, where they can use radio frequency waves to create covert communication techniques where you don't need a phone anymore. They can just beam information straight to your head. That's a literal patent for that. I can show you if you'd like. I'll just show you the patents real quick.
Before you jump into that, let me just show one thing real quick so people understand about the sound that I've had pulled up for a minute. If you wonder why I have dropped Nikola Tesla's name a couple times in this, this maybe will help people understand. I'm not going to share
You want me to drop share?
the sound. Yeah, I'm not going to share the sound on my end. I'm just going to show you kind of what I mean. I've had the same avatar in our back comms since almost the beginning. Not many people have asked me what it is, which is fine, but this is, I think, an easy way to understand what sound is capable of. And again, I don't really need the music here because I don't need the copyright strike. And then they'll use it on multiple. Like I said, I'll kind of see there's what they're using of the drums. But I'll fast forward to Tesla's Coil, which is right here, I believe. Yeah. So they put on these metal suits now. Like what you would probably see is like a shark suit. There's it being converted into energy. So I find it very interesting, and that's why I kept dropping Tesla's name a couple of times. And I'll just scroll up for a second to show you this. It says, did you notice that the Tesla coil, Nikola Tesla, clearly knew about somatics back in the 1890s? It's defined as the study of how sound affects matter. And this was a famous quote by him. If you wish to understand the universe, think of energy, frequency, and vibration. And that's why I said that about why they bankrupted him. J.P. Morgan and the likes were funding him, and they removed his funding. And from what I can understand, left him broke until the day he died. And then all of his studies, all of his papers, apparently was from what I understand was taken by, well, some people report say John Trump, was that Donald Trump's uncle? They'd sent in there to retrieve his papers. So I just thought that was interesting. And you can carry on. I just kind of thought that was, and I will send that to you for sure. I'll drop it in the chat.
Thank you.
I love this website, by the way. It goes through all of it. It goes through that. It goes through the symbology, which is what I get into. But yeah, carry on with the next one.
All right. Yeah, I'm excited to go browse that. So voice to skull technology, it's the radio frequency hearing effect. And we were talking about ultrasound interacting with brain cells after they deliver genetic payload through something like a virus. And here's one of the 2 from the Air Force that discusses using the thermoacoustic effect to transmit speech using radio frequencies. And it's the radio frequency hearing effect. It's a very known thing. And radio frequency effect is explained and analyzed as a thermal to acoustic demodulating process. Energy absorption in a medium such as the head causes mechanical expansion and contraction, and thus an acoustic signal, which means sound waves. And that's with wireless energy. What we understand is what cell phones do, and it can create ultrasound, mechanical sound waves, which is what ultrasound is. They're sound waves. They're just at a frequency higher than 20,000 hertz. So if you use microwave energy or millimeter wave energy, could you deliver ultrasonic energy pulses in the body after it converts to a mechanical wave? Using electromagnetic waves as it passes through the body that converts into mechanical waves, could you pulse the energy in such a way to have it be an ultrasound, something higher than audible hearing, but still be mechanical in nature? And I submit to you the answer is yes. So there's a few ways, so to speak, that wireless networks can be implemented to interact with human biology. So that's sonogenetics. Nanotechnology, piezoelectric nanoparticles. This is where it gets weird, man. Or another layer. The assessment of the effects of a wireless neural stimulation mediated by piezoelectric nanoparticles. This is I have to pay for this article, so I can't read it, but there are other links that kind of support this. Wireless brain stimulation, neuronal stimulation, mediated by ultrasound and piezoelectric nanoparticles represents an unprecedented approach aimed at cell activation. Recently, we demonstrated that barium titanate nanoparticles, barium, you say, huh? Isn't that one of the things that they like to spray up in the air? Barium, strontium, and aluminum, barium salt, strontium, and aluminum. So barium nanoparticles behave as excellent nanotransducers by eliciting specific cell response following treatment with ultrasounds. In this chapter, we describe in detail the techniques exploited to investigate nanoparticle cell interactions and the activation of neuronal-like clusters in terms of sodium and calcium fluxes. These are the electrolytes in your body, the things that regulate the voltage in your body. We are electrochemical. And if you know what electrolytes are, they're needed. If you don't have enough, you die. If you have too many, you die. And this is what regulates the voltage in our bodies. Some of the ions, sodium and
And I
calcium.
think that's why they attack our systems on all fronts, from the food that they grow in the ground to what we breathe in our atmosphere to the water that we drink full of fluoride. It's
yeah.
Okay.
And here's another one that may be another example here. Acoustic stimulation can induce selective neural network response by piezoelectric nanoparticles. The reason I'm kind of sharing both is to give you the biologic and also the nanotech piece. We're past the nanotech, the synthetic nanotech piece. It's been done. It's already been done. And that's where I think we're already pretty much on the internet already. And a lot of people may not agree with me, which is fine. You're entitled to your opinion, but we're moving into the age where they're dealing with genetic expression, where they're going to use your own body to grow this stuff so you can't get rid of it. Right now, you could take heavy metal cleansers potentially and get rid of a lot of this nanotech. Ultrasonic wave generation by time-gated microwaves. Now, this article, going back to the voice-to-skull technology, the thermoacoustic effect, and this idea of creating mechanical waves with microwaves, this document, Journal of Acoustical Society America, ultrasonic wave generation by time-gated microwaves, shows how energy transfer between electromagnetic waves and elastic waves at the surface of a fluid or a solid material could constitute a new way to generate ultrasonic waves without contact for the purpose of non-destructive evaluation and control. Now, they're talking about looking at materials to understand things like, do they have cracks in it? What's their density? But the looking at the fundamental physics of what's happening here. This is a document that discusses using electromagnetic waves to generate ultrasonic waves as they pass through a medium change, going from a gaseous state to a plasma state or to a solid state or to some hybridization of that, because that's what we are. We're plasma, we're water, we're solid bone. And that's, you need the transition, the medium transition, to go from air to something else, or from plasma to a bone, or a bone to a water, or a bone to a plasma, a bone to tissue. When you get the change in the property type, the material type, you get the opportunity to generate pressure waves. You get vibrations, and they can do this by generating electromagnetic waves.
And that's
So-
and the way to do that is because of the time, dilation, or the gating that you're talking about between the different mediums, because they have to say, okay, well, we're going to have this go through this medium, and so based upon their gating, or the way that they kind of the bursts, they can to a particular area.
Mm-hmm. Yeah, they can map it all because they can read back the vibrations. So think of it like radar. You can send out pulses to create mechanical waves in tissue, for example.
And you get feedback too.
Correct. You can send subsequent waves to then measure where the dielectric properties, if you will, how they've changed, where the densities are and where they were, and all of the other subsets that they can measure with an electromagnetic signal. So they can use the electromagnetic signal to create a change and then use the same transmission device, the antenna array, if you will, to then monitor what it is they're doing and make on-the-spot corrections. And it's all done with algorithms and machine learning. Thermoacoustic imaging, it's a very real thing. They use it for mammograms and stuff. Patents in here for stimulating neurons. And now we get into mechanogenetics.
I would assume that most of the audience would maybe just, can we go over something quick with voice to skull, because I'm sure that's like it's more of a buzzword that people might know.
We I showed a patent for it a second ago, which is fine. We'll go through it.
Well, sorry, if you already covered it, I had to change devices, so I didn't know if we already went through it.
You're right. Let's elaborate a little bit. This might not be a good one. Yeah, no, not this one. There's another one in here. This might be it.
In San Diego, California, Woody Norris, developer of the LRAD, describes another of his acoustic technologies known as hypersonic sound.
It's kind of like radio stations, except in this case, you don't need a receiver. To hear a radio station, I got to have a receiver. With this, the mixing actually happens in the air, and you hear it without any other device. So if I aim it at you, and it happens to hit both of your ears, and I'm reluctant to say this, it'll be as though the sound is inside your head. This time I'll come all the way around in case you're getting a lot of echo from the room. Much of it is people pleading for help because they've been hearing voices in their head for years. And now this technology explains it. If it's my sound, turn your head, plug your ears, cover your head, sound goes away. If it doesn't, it's not me.
The observation of the phenomena came way back during the Second World War, and the radar operators, accidentally noticed that when they stand in front of the radar, sometimes they can hear clicks in their head. Somebody by the name of Alan Fry, who had talked to some of the radar operators based on their report, and he conducted a very simple laboratory experiment, indeed showed that human beings can hear the radar pulses. They were not just making it up.
They wanted to know what the significance of something like this would be if man had another sensory motor input. And of course, the possibilities are staggering. Like you have another sense like sight, only you're not using it. What do you think that would imply? It means you're walking around being blind to something that's constantly giving you inputs, but you're not aware of it. Well, the first implication on that would be a weapon.
There was some concerns because the American embassy in Moscow was reported to have been bombarded by microwaves. So the speculation was that, are they trying to gather intelligence, or are they trying to actually mess with the brains of the embassy personnel? So there was some experiments being conducted. Of course, then they were involved in some other things which I'm not involved in, which are classified in Nature.
Dr. Lin configured the equipment for the 1973 experiment performed at the Walter Reed Army Research Institute, wherein the experimenters successfully encoded speech and transmitted it via microwaves. The subject of the experiment, Dr. Joseph C. Sharp, was able to hear the spoken words for 1 through 10, and the results were published in the Journal of the American Psychological Association. And while Dr. Sharp verified these results during a personal interview with the filmmakers, he declined to have his voice or image recorded, stating that the technology was still too controversial.
In ordinary sound perception, sound that goes into the ear canal gets amplified by the small bones in the middle ear and then gets to the inner ear where the cochlea resides, and in the cochlea is converted to electrical impulse. But fundamentally, the physical phenomena that enables it is mechanical movement of particles. So the mechanical wave transmitted by the bones of the middle ear gets converted at the inner ear into the electrical impulses. But in the case of a microwave auditory effect, is the source is electromagnetic. So if you expose the biological tissue to a pulse of microwave energy, the tissue expands and induces a vibration. Instead of going through the middle ear, the microwave-induced vibration gets propagated to the inner ear directly, and at the inner ear is converted to electrical impulses again. And in this case, a vibration in the human tissue, if the tissue is the head, for example, will fall within the auditory range of a human subject.
This is like another sensory motor input that man has not yet learned how to use.
Of course, you could, as I say, if you have a high enough power system, you can have a group of people subjected to the same type of a phenomena.
There's all kinds of applications there, you know, in terms of making someone think they're crazy. To hear voices in your head, what do you think that's about? If you started hearing voices in your head and it wasn't your voice.
I don't know whether it is true or not. I think there's a sizable number of people who think this is being used for mind control.
Mind control is right at the top of the list. That obviously is what everybody would like to do, is control everybody's opinion on something, right?
I think if somebody who wants to do it, there is the potential.
Good. Kenyatta?
We just heard about something similar to this in Cuba, remember?
Yeah.
The same thing he was talking about, the embassy in Russia. That's exactly what was going on with the embassy in Cuba, our embassy in Cuba.
Yeah. So you've got lots of patents here. If you guys want to, we're not doing it today, but acoustic signals from neural timing difference data, applying ultrasonic acoustic data to the human neural cortex, device and method for non-invasive neuromodulation, device and method for modulating brain activity, methods and devices for modulating cellular activity using ultrasound, method and system for direct communication. This uses ultrasound to directly communicate. So we're going to transition, if you're ready, to another genetic type, similar in nature. It's Mechanogenetics. This is where they can get your cells to be more sensitive to sound. Acoustic Mechanogenetics. And what it is, it's the same thing, right? Where you have tissue modulation, gene activation, gene delivery. So they use a viral vector or otherwise to deliver genetic information to make you way more susceptible to ultrasound. And it's an actual genetic, and I can't access this. It's pay to play. But if you go to these things, you get all kinds of rabbit trails. For example, over here, molecular and cellular engineering gene therapy. Download PDF for testing thousands of nanoparticles in the body using something. We're not going to go through it, but there's more on Cas9. Cas9 mediated cell differentiation. So that means that they can go in and selectively target which cells they want to deliver genetic information too. That's what they're talking about there. Just, so we're we're not in Kansas anymore, man. We are living in crazy sci-fi land. Mechanogenetics for remote and non-invasive control of immunotherapy. A mechanoreceptor. Let's look and see what this is. Mechanoreceptors. Now these are in we have these all over the place. Mechanoreceptors, the things that they would be able to manipulate to be more responsive to sound, which also means that the normal ones also are responsive to sound. Detect stimuli such as touch, pressure, vibration, and sound from external and internal environments. They contain primary sensory neurons that respond to changes in mechanical displacement. So you have these, I mean, that's your sense of touch. Basically, your tactile sensations are a lot of them are mechanoreceptors. There's also thermoreceptors, which deal with temperature. They can introduce genetic information to manipulate that. Ultrasonic control of brain activity through the activation of mechanosensitive channels. Impulse and pressure waves cause excitement and conduction in the nervous system. Going back to that voice to skull stuff, it's beyond just being able to hear things. If they can induce pressure waves, then they can start tweaking on your nervous system too. It's not just
That's
your
why
ears.
I played that short video of how it could manipulate water. And what are we mostly made up of?
Yeah, water. Nerve excitement. Conduction is caused by voltage changes. However, the influx of fluid in an elastical tube releases impulses or pressure waves. Therefore, an influx of ion currents can affect elastic neuronal cells and fibers, including and induce impulses. Impulse waves can have a high speed. It goes on and on. Talks about the myelin sheaths in your nerves and different sensory cells and lipid membranes of the cell. And I got to read this again. This one looks really interesting, as a matter of fact. So thermogenetics. We'll get to the chimeras, the chimeric animals in a minute, just to see, put a nice round bookend on it for you. Thermogenetics, using temperature sensitive channels to study brain circuits. Neuroscientists develop potential tools for the study of brain function, thermogenetic neurostimulation with single cell resolution. What? Thermogenetic neurostimulation, single cell resolution. It's a promising, innovative neurostimulation technique which enables robust activation of neurons using thermosensitive, that's temperature change, a receptor potential. It's a TRP receptor, cation channel. So it's an ion channel. The common denominator here are these ions, the sodium, potassium, calcium, and others that are ions. They're positively or negatively charged ions that regulate voltage in your brain and your nervous system. And they have channels that then allow the positive and negative to flow back and forth. And that happens when a neuron fires. So everything we've been looking at, or most of what we've been looking at, has to deal with these specific types of these ion channels. And in this case, these are ones that respond to heat. And they can go in and they can manipulate this stuff. And they talk about it here. They set the foundation with optogenetics, that manipulation with light. They allow specific neurons to be operated with light, with high precision in space and time. They're helpful in understanding mechanisms and functioning of the brain. So thermogenetics offers a promising alternative approach based on heat activation, cation channels from the TRP family. There's a whole family of these things that respond to heat. So heat, you touch a stove, you pull your hand away. You put your hand over a lighter and it hurts, right? You touch an exhaust pipe, that kind of stuff. TRP channels in 3 orders of magnitude, more conductive, can be activated by heat induced with infrared or magnetic particles. Those are just examples. So infrared, right? We know about that. Infrared, that's how your Face ID works on your phones. magnetic nanoparticles, magnetic particles, well, that's a whole other topic. And you want to look up a lady by the name of Polina Anikiva. She works in MIT. She has her own lab, and she works on nanomagnetic particles that do exactly that. Multiple magnetic nanoparticles delivered to the brain, so that when the brain is in a magnetic field, a changing magnetic field, they're able to turn some on and others off, and create complex bidirectional communication with the brain using many different types of magnetic nanoparticles that are all delivered at the same time. And that's a very real thing, and there's documents in here that discuss it. Mechanogenetics and thermogenetics for immunotherapy, acoustic and ultrasound-based mechanogenetics and thermogenetics. So what else is in here? Wow, we're on 44. Optogenetics. So this is the idea of combining these things, combining sound and light, where you use nanoparticles that are responsive to sound, which when they're excited by the ultrasound, they emit light. So you can use ultrasound to manipulate cells, but you can also use the same ultrasound to excite nanoparticles, which then emit photons of light that also manipulate cells. And you have these things called mechanoluminescent nanoparticles, which are these things that when you excite them with sound, they emit light. These are very real things, mechanoluminescent nanoparticles. Certain nanoparticles exhibit intense light emissions during their elastic deformation, called the phenomena of elastomechanoluminescence, and they give you some compounds. Nanoparticles are deformed in the elastic region by stress, initially increasing with time, and then attaining a fixed value, or by the stress initially increasing and decreasing with time. So, who knows what they're putting in us at this point? The point of this is that there are many ways this can be done. Mechanically excited multicolor luminescence lanthide ions. I don't know what time. Yeah, it's 9:30. We need to keep moving. And then you have magnetogenetics. Magnetogenetics. Let's find. I want to find a particular video. I'm hoping that it's in here. Magnetic yeast, magnetoneuron project. This is idea of growing ion, magnetic responsive ions. Basically, you're growing ferrite in cells, which is iron. You naturally produce this stuff in your body, and I'm trying to find it. It's from Harvard. I don't think it's here. That's very interesting. It was my favorite. It's one of my favorite videos, actually. Let's see if we can find it real fast, because they literally talk about being able to remotely control the brain with magnetic fields. Well, we're going to give this a quick shot and see if we can find it. If you ever want to go down a rabbit hole, go to the Wyss Institute. The stuff they have here is just ridiculous.
Wait till you start reading the narrow gate. It'll blow your mind as well.
It's not here.
No results found. That means
okay.
buried.
Yeah,
Trying to hide it from you.
It's all right. It's not something that Look at this. RF smart tools, RFID tracking for surgical instruments, DNA data storage. This stuff is crazy. Multifunctional, multi-scale, reconfigurable. Let's see if we can go down and find some related stuff here. Microparticles. Yeah, we're gonna get lost in translation here. It's all right. I'll try to look for it and find it at another time. Just know that they can grow this stuff in your body. There it is. Is this what it is? Magnetic yeast. This is it. Look at this girl. Look at her face. Is that not crazy?
Mm-hmm.
Maybe she's a nice person. I don't know, but she kind of freaks me out.
Oh, I've got something on this too. It's crazy you're bringing about magnetic.
So magnetism in nature is an interesting phenomena. There are organisms that, of course, use the magnetic force of the Earth in their migratory behavior. An interesting example are actually monarch butterflies. And that's an interface between their antenna and the magnetic field of the Earth. What's unique about our work is that we have created a cell that will migrate towards a magnet.
Our study is the first demonstration that to make a naturally non-magnetic organism to magnetic.
What is partly the motivation behind our work is bacteria that are magnetic, and it's not clear why they're magnetic. It may be sort of an evolutionary artifact, but they can align themselves with the magnetic field of the Earth, and as humans we can actually find this to be very useful in terms of remediation and things like that. So first of all, the basis for the magnetism is the uptake of iron by the cells, so iron can become magnetic. And so the cells will naturally take up iron, but normally they will sequester it inside a compartment in the cell that's called an organelle. And when it's sequestered, the cells are not very magnetic. So one thing we did was to eliminate and inhibit the normal ability of cells to hide the iron and to make it more exposed. The second is to increase the uptake and the crystallization of iron to make it magnetic. And the third is to manipulate, to fool the cells into creating an environment where the iron will be more magnetic. Well, my favorite experiment that we did, and when I say we, I really mean KG, is that he could take the cells and put them in a dish and then hold a magnet underneath the dish, and the cells would migrate towards the magnet, and he could have them make patterns in the dish depending on where the magnet was. And so one of the things he did was have them make a pattern of a face. There's the potential medical applications. There's the potential for applications in bioprocessing. There's potential applications for tissue engineering. It could have particular importance in bioprocessing. So you have large vats of cells, and sometimes you need to pull out either the bad cells or the good cells. And so you could imagine if you had a giant magnet, you could suck out the good cells from a bioprocessor.
It could be also possible that you may be able to stimulate cells using external magnets and engineer cells to respond.
One of the interests is how do you make living forms interface with machines? So you want to be able to have cells that can communicate with machines. And so this is a perfect example of that. You can have a magnet and the cell can be communicating with it. So the magnet could be a form of input, the magnetism, and then the cell does something. And so this interface of life with machines is part of our future.
I think that last piece kind of speaks for itself.
Mm-hmm. It's amazing how much they've hid from us, though. How they tell us, oh, this new discovery. Yeah, not so new.
So another combination, magnetothermal genetics. It's just, the recipes are endless. So your guess is as good as mine. What's being used, what's going to be used. Another way to hack into the brain to control physical behavior in mice, magnetothermal genetics, using magnets and nanotech to rapidly heat up specific parts of the brain to get the thermoreceptors to respond. And this is more like what I see kind of already happening. This with the piezoelectric transducers. This is the kind of stuff I see them putting in food, water, air. So we're going to move forward. I know we're probably sitting on, where are we at, like an hour and a half already?
Probably, yeah. And we'll quickly hit the Chimeras, and there's 3 stories in this. Quick little brief paragraphs kind of tie into what you've been talking about. The things again on that website that I love to utilize, the narrow gate, that may blow some people's minds of the things that's been discovered and been suppressed from us as well.
Yeah. Here's another institute. Remote control of gene expression, Magnetogenetics, Rockefeller University. Combining nanotechnology and bioengineering to demonstrate externally and internally genetically encoded nanoparticles. Genetically encoded nanoparticles can be used in the body to remotely regulate cellular activity. So this stuff is very, 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 very real. And you have electrogenetics, where they can get cells to be more responsive to electric fields or electric stimulation. And you have chemogenetics, being able to use designer chemicals to manipulate brains and the nervous system. That's a very, very interesting field to research as well. The program for chemogenetics that they're used is a system called DREAD, ironically enough. And when we get, it's where we get into SARS and COVID and PCR testing and gene editors for rapid testing. DARPA program, gene editors could find a new use for rapid detectors for pathogenic threats. Biosecurity tools from IARPA. IARPA wants to protect the nation from genome editing. So these people are all into this stuff. All into it. And here we are. They've immersed the world in a reason to play God. And then we get into organs on chips, how they can experiment on human cells without necessarily working on humans. Organ on a chip. It's a whole nother topic. We could do a whole show on just organs on chips. It's a ridiculous field.
Yeah.
What else is in here?
So,
this is where we get into nanotech and Polina Anikova, neural interfaces. Facebook's mind reading chief quits, gets into big tech and their look at brain computer interfaces. Peripheral nerve interfaces and prosthetics from DARPA. Anyway, I think we're ready to move on. I want to show you this real quick before we go. This is a patent from MIT, I believe. Talks about vaccine nanotechnology. It'll give you a good picture of just a rough outline of how sophisticated this stuff is. And this is just a basic example. They don't really give you super complex examples in patents. They just give you a basic example, if it'll load. If any day now. Any day now.
That's interesting.
Yeah, it we'll let that spin for a minute.
Doesn't want to give it to you.
No. Yeah, we could do a whole show on nanotech. There's just, you see the links here. It's endless. Nanowires, nanotech. Oh, nope. There we go. Vaccine nanotechnology, Harvard College, Brigham and Women's Hospital, Children's Medical Center, Massachusetts Institute of Technology, Partners Healthcare Systems, and everybody's got it. Provides compositions and systems for delivery of nanocarriers to cells of the immune system. Provides vaccine nanocarriers capable of stimulating an immune response in T cells or B cells. I'm going to show you some of these pictures here. And there's one in particular. It's really this. They can design this stuff. It's like Legos for smart people, or Tinker Toys for smart people, where you have a lipid, fat, that you make a ball out of, a hollow ball, and these are specific receptor types that are looking for specific markers in the body. Same thing with these. They do different things. They can be used as an if-then statement. So if something triggers this, then this will become active, or vice versa. And inside you have something like a genetic payload or medication or anything. So what this thing is a completely designed from the ground up vehicle that is able to go in and be stable in the body, find a specific place to go, and then do something, perform a specific function, completely synthetic from the ground up, is really what this thing is. There's 54 images that this vaccine patent has. There's a couple more of these cool little dots here.
Yeah, I completely agree. The patents will blow people's minds. Why do they need a patent for it if they're not testing it or running experiments on it?
Yeah, they could very well have good applications, targeted therapies for cancer and genetic mutations. So you can go in and fix somebody's genetics. I get the benefit of it, but we're a month away, 2 months away. Your guess is as good as mine from military deployment of genetically modifying our entire society, if it's not stopped. And what you'll see in these variations is just different constructs of different types of nano vaccines, man. All of these are different components that can be stabilized in the body and delivered in sequence. And this stuff is freaky, man. It's just really scary that we're here and the regulations on it are basically there is none. There's an open ticket to screw with the human species at this point. So
yeah, it's scary for sure.
Let's I think we're good with the groundwork. Let's look at some of these human animal chimeras, biologic research and ethical issues. This is back in 2016.
And before we get into that, let me steal the screen share from you really quick, because I just want to go over these three things. They're pretty quick, but it kind of highlights what you were talking about. And again, I found this on the link I dropped you, the narrow gate. This starts with Nikola Tesla, and it says Tesla invented a medium for wireless transportation of electricity, i.e. the Wardenclyffe Tower, pictured below, by understanding the natural resonance resonant frequency of the Earth. Unfortunately, the powers that be, in this case Tesla's financiers J.P. Morgan and Westinghouse, pulled all funding, and he was kept broke and isolated in a hotel room for the rest of his life, as there was no way for wireless electricity or many of his other brilliant inventions to be monetized. For anyone interested in learning more about this story, there's a biography which I actually have pulled, ready to buy, to read. This is, I don't know, have you ever heard of the American inventor by the name of Dr. Royal Raymond Rife? From 1888 until 1971, found that pathogens, bacteria, and viruses, including cancer, each has a resonant frequency, and he was able to target and destroy these cells by means of an electronic resonator. Needless to say, he too was shut down by the big money pharmaceutical powers, as there's no money to be made from curing people. I highly recommend reading more about the Royal Rife and his invention, perhaps starting with this article and the website. Also, this is what I like about this website. It links you to other places to read about this, so you can check this for yourself, do your own research. It's so I'll make sure to link this below too, but this is a picture of him with this powerful microscope where he could target the cells. This is another man. You was talking about magnetism. This goes in a little different direction, but just shows you what you can do with it. Another person who discovered the secrets of resonance was Ed Liskinen, I'm probably saying that wrong, sorry, who built the Coral Castle by moving huge blocks of limestone using only a harmonic oscillator based on magnetism. There are plenty of videos on YouTube about him, etc., etc. This is the little city, and this is him with that machine, magnetic current. So these are all things that, you know, have you ever heard of these? I know you've heard of the first one, but have you heard of these last two? TMC? Anybody? Have you ever, the last two of those people mentioned? I hadn't until I read this.
The limestone, yes. That
I know.
the coral, yeah, I've seen that one. I've heard of that one. They still today they don't know how he did it.
Yeah, exactly. And then you can go ahead, TMC, and bring that. We'll quickly hit the human chimeras because we've been going for a while now. But I think that he set the stage for helping you understand. I'll show this one quickly. Another article, I think he's going to bring it up as well. Hit quickly on what they're talking about here. They're a further example of an interspecies chimera generated when human cells are introduced into animals. This can be done through a variety of techniques. The part that I find interesting is where it says these human animal chimeras are also produced by introducing human cells into animals during various developmental stages, be it embryonic, fetal, or postnatal after birth, according to a 2007 article. And you're like, whoa, 2007, that's nothing. Because I'll highlight this part where it says, I lost my spot, where it's, there it goes. It says for human embryos, which can also be engineered in the lab. Well, we know that because women go all the time and have assisted, whenever they have issues getting pregnant, they create those embryos in that saucer, and they, inject them into the woman's womb. And that's how, when women have fertility issues, they give her the opportunity to give birth. This is what they tell us in order to help. They don't tell us what's going on in the back rooms when they're generating these human embryos. But this is, I'll just mention this one briefly, where it says such research has been conducted for decades. Now, for instance, in 1974, a group of researchers from Denmark reported the first successful transplantation of many different human fetal organs into a laboratory mouse model called the nude mouse. This is in 1974. Showed that human fetal lungs, kidneys, pancreas, all this, the ovaries themselves developed within the nude mouse in 1974. So if you think that this hasn't been done and hasn't been accomplished, they've been doing this for what, almost 5 decades that we know about.
Yep.
And so, and it's funny because you mentioned, I'll mention this one either, and might be something else people are not aware of. This is, for people wondering, this is at a time. It talks about it. This is in 2016. It says nearly a year ago, the National Institute of Health decided to stop funding research in a promising area of stem cell science. Scientists eager to find new ways to generate desperately needed human organs or tissues to replace damaged or diseased ones have been excited by the possibility of inserting a specific type of human stem cell, which has the potential to turn into any type of tissue into animals, where they could develop and eventually be transplanted into humans here. But the agency now says that it will lift the ban and put in place a review process that would require 2 types of chimera studies to get further review. These include experiments in which human stem cells are added to very early embryos of other animals, and studies in which the human stem cells are injected into the brains of mammals. What are we other than rats and mice? Notice how it just says mammals.
And it says other than rats and mice.
Exactly. That's why you see, this is how they get around it. They just put mammals. That covers us. So, I mean, again, these are just, it's telling you right here, this has been done. And this is the one, what are they trying Genetics closest to? First human monkey chimeras raised concern among scientists. This is from August of 2019. So do you think these people care? Do you think they care about what they're doing? Well, it's all experiments to them. I think they started doing it in China. Yeah, the research was conducted in China to avoid legal issues, according to the report. And if you want to go down a rabbit hole, and this is just me talking here, I know that people know about China's organ harvesting. And I know they know about the Falun Gong practitioners and Muslims, where they round these people up. That's why you can basically get organs on demand within 2 weeks. They can match your blood type and size in 2 weeks. And again, my belief is, you see how long they've been doing this. Do I believe they're just doing this with people that they're taking and harvesting from? My personal belief is they saw a cash cow in this years ago. And if you think China has any issues creating human embryos to develop organs that people will buy on demand, I think you're living in a dream world. But yeah, I believe it's probably even gotten that far, my personal opinion. Because if we're learning about this stuff today, that means they've been probably doing it for decades. Just from all the things that we've seen. And I'll shut up and let you add to it, because I know we've been going for a while. But I just find it just find the ties are kind of there.
Yeah, we'll look real quick at these articles, but I think we're at a good stopping point, just to round out this idea of humans manipulating genetics. And what does it mean? And it doesn't stop in any one category. It's not just crossing animal species with other animals. It has to do with going in and making very sophisticated upgrades or changes to existing genetic lines. Here's another one. I don't think we should read all of these. 3 human chimeras that actually already exist. Human animal chimeras. 2 sets of DNA with the code to make up 2 separate organisms. One way chimeras can happen naturally in humans is that a fetus can absorb its twin. This can occur with fraternal twins if one embryo dies very early in pregnancy. Some of the cells are absorbed by the other twin. The remaining fetus will have 2 sets of cells.
Yeah, then they're not talking about this as this is a possibility. They're telling you they've done this and this is what's happened.
Person can also be a chimera if they undergo a bone marrow transplant. These transplants of these type occur when treating leukemia. They'll have their own bone marrow destroyed and replaced with bone marrow from another person. Bone marrow contains stem cells that develop into red blood cells. This means that a person with bone marrow transplant will have blood cells for the rest of their life that are genetically identical to those of the donor and are not genetically the same as the other cells of their own body. That's why it says in some cases, all of the blood cells in a person who received a bone marrow transplant will match the DNA of their donor. In other cases, the recipient may have a mix of their own and the donor. This is back in 2004.
Yeah, there's no real pushback on this. There's the article I just mentioned, regulations were lifted to continue this study. They were doing it in other countries that, had no ethical concerns. Showed you the article from the 70s where they were talking about already doing this. And again, we're in 2020 now.
Yeah, these are somewhat, I don't know, I won't call them natural, but these are a little more, I guess, easy to digest. The third way really is, I think, going to really resonate with everybody. They said in 2015, a study suggested that all pregnant women, at least temporarily, right, basically have their children's genetics floating around in their bodies for a period of time. The researchers tested tissue samples from kidneys, livers, spleens, lungs, hearts, and brains of 26 women who tragically died while pregnant or within 1 month of giving birth. The study found the women had fetal cells in all of their tissues: kidneys, liver, spleen, lungs, heart, and brains. Had fetal cells there. The researchers knew that the cells were from the fetuses and not from the mother because the cells contained a Y chromosome found only in males, and the women had all been carrying sons. In some cases, fetal cells may stay in a woman's body for years. In 2012, a study, researchers analyzed the brain of 59 women ages 32 to 101 after they died. They found 63% of the women had traces of male DNA from fetal cells in their brains. The oldest woman to have fetal cells in her brain was 94, suggesting that these cells can sometimes stay in the body for a lifetime. And this really ties into this idea of introducing genetic information into the body. And we're seeing here that in some cases, genetic information stays forever or stays for decades or more.
Yeah, this is when you stop having ethical concerns and are just trying to be the first one to, the fastest to the finish line without thinking about the consequences of what you're doing.
Human animal chimeras fast on track. This is in 2019.
Yeah, again, remember, this is what they're telling us. And TMC, from your experience and everybody else in this room, and whenever it comes even to the military or DARPA, do you think we learn about something like LED lights? Do you think LED lights came out the year they showed them to us?
No.
Or it was something they were using on military aircraft for 40 years?
Yeah, I got a feeling that DARPA, there's many layers to DARPA. The stuff they tell us about just kind of justifies the public expenditure. But then I think there's groups that are above even DARPA where they feed DARPA information, so to speak, and say this is where society's going. So here you go, and out they go with it, right? They're able to set the tempo for what needs to be researched and focused on and studied. It's just my opinion. I don't think DARPA is the top of the food chain. That's all I'm saying.
I would agree completely. Go ahead, Breaking, sir.
I was going to say, and to that end, though, China, if you recall, had dabbled with CRISPR technology, and I think it was January 21st, 2019. I've got an article here. Death penalty likely for Chinese scientist who created world's first gene-edited babies. That was out of the Mind Unleashed back then, and I think that only happened because there was enough international outrage brought about by it. And that being said, what's to say that these guys haven't been doing this all along and we just now found out about it?
Yep.
That video you dropped in there, I was trying to scan back.
That was part of that article.
Yeah, I was trying to scan back. That's what I was talking about. Dr. He, that the little bit of pushback he got. Yep.
And here's one. Human animal chimera studies will soon be allowed in the U.S. This was back in 2016.
Yeah, that's when I pulled from and read the where the second paragraph. Yeah, about how they're going to allow it again, which to me is comical when they say, "Oh, we're opening it back up to studies. Never mind that secret, science lab that you don't know about. It's been doing it all along." I mean, look at Area 51. I mean, just to take for example, or is anyone allowed to go in and out of there? I mean, do they not? What do they think's going on in there? Development of different ice cream flavors? I mean, really? They don't care about the laws. They don't care about ethics. Care about any of that.
I guess we'll finish up with this. This is the President's Council on Bioethics. There's a couple links here that we'll look at. And I encourage you to go look at this yourself because this literally discusses patenting humans. Embryo Project Encyclopedia of the U.S. President's Council on Bioethics. Human stem cell research. So this is this is a subset of it, but this is the actual archive. Bioethics Archives in Georgetown. And being human, President's Council on Bioethics. Okay, we'll start at the home page here. Topics of concern. This was founded in 2001 and it was disbanded in 2009.
Were you going to say something breaking? I saw him unmute, and usually he does that. Do you have something to add?
I'll wait till he's done on this part.
Oh, okay.
Provide adequate moral and ethical lens through which to view particular developments in the proper scope and depth. They're talking about bioethics or biotechnology. Advance the human good, avoid harm. Biotechnology must be used within ethical constraints. And task the bioethics to help society develop those constraints. Doctor. The topics of concern: age retardation, life extension, aging and end of life, beyond therapy, meaning enhancements, human enhancement, biotechnology, public policy, bioethic commissions, bioethics and literature, cloning, children, death, drugs, children and behavior control, genetics, healthcare, human dignity, memory boosting or memory suppression, mood control, nanotechnology, neuroethics, newborn screening, organ transplantation, professions, property in the body. We'll look at that briefly. Research ethics, sex selection, and stem cells. The property in the body piece is where I wanted to go briefly. The patentability of human organisms. There were lots of discussions about the patentability of human organisms, one of which I read pertaining to this idea of stem cell harvesting, whereby you go in for in vitro, you get 7 to 12 samples that are fertilized and then flash frozen. Once fertilized, the in vitro is a success with the first implantation of 2 or 3, and the rest basically are up for sale. And then we get into this idea of patenting people, patenting stem cells. And where does it stop? Does it stop at the embryo, at a child, at an adult? If the adults patent, does that mean their children are also subsequently patent? They actually go through these types of discussions in these hyperlinks here. And all you got to do is search Patent Human President's Council on Bioethics. President's Council on Bioethics, and you will be led, the second link here will take you to one of the sub pages whereby if you go over to the home page, you get this page right here, and there are links that you can pick to your heart's content. You want to look at cloning, you can look at cloning real quick, and then human procreation and biotechnology, ethical issues on clonal reproduction, policy issues on research and cloning, the proper use of language. This was 2001 to 2009.
Well, it's interesting that you chose like a couple of those, and there is sex selection, and the what was it, behavioral control, and then we see what's going on in the world today with all these kids that are confused about who they are, what gender they are. I mean,
prescription stimulants used in American children, neuropsychopharmacology and public policy, pediatric psychopharmacology, staff background paper, human flourishing, performance enhancement, and Ritalin. President's Council on Bioethics.
Yeah, I found the link. I was hijacking it from the top. I was reading, so I've got that link. I'll put it down in the description as well.
Memory boosting and suppression. Where is that one? Is that over here?
Thinking this came from Georgetown as well.
This is archived at Georgetown, yeah. Remembering and forgetting. Beyond therapy, better memories. staff working paper on better memories, the promise and perils of pharmacological interventions.
What is better memories? Oh, I mean, you know what I mean? What's it? There's authentic memories. Is there better memories? What does that mean?
Yeah.
They're going to implant better memories into your head? That's.
Why should we have to live with painful, guilty, shame-filled, shocking, or unpleasant memories if we don't have to? And science could offer us a way out. That's a question that Gill offers a kind of argument for, an argument for which he can make for himself, or we can draw on if we wish. But I think that's the question that this paper is trying to get us to think about.
Is that like bleach bit for the brain?
Yeah, it's like going in and rewiring it or doing a chemical lobotomy on it.
There's the part that they're leaving out there. If they can insert memories, then that means they can remove them.
Yeah, and DARPA has entire programs where they can do that electronically at this point. This is back in the time when they were looking at using pharmaceuticals to do it. Now we're at the point of electromedicine where you can use chips and you can use electronics, wireless energy interfaces to do the same thing in a much more targeted way. And so nothing's changed with the intent. It's just the tools by which these things are being achieved is really all that's been changed. But you can go in and read all these are literally like minutes that were taken where they were having open discussions on this presidential council, and it was totally all of it was documented, and now it's archived. You can go
and those two names are sticking out to me. Other than this that you're bringing up too, especially that Cass. That name Cass is ringing a bell, and I can't remember. I'll have to dig into that because I remember that name. Do you remember that breaking Cass?
You're thinking of Cass Sunstein?
I don't know. It
doesn't ring a bell here.
I don't know. Okay.
No, the only one that does is Cass Sunstein. That's the regulatory czar for Obama.
Anyway, I guess I'll drop share.
Well, while you do that, the one thing that you touched upon there about DARPA and whether or not they are the ones giving the edicts or if they take orders, what have you. I want you to think back to, say, January, February this year as this quote-unquote pandemic started off, right? There's a lot of finger pointing, wet markets, bats, whatnot, that just so happened to be, a stone's throw from a particular lab in China and Wuhan. And I also want you to think, if you remember seeing the pictures of the signage outside of the Wuhan virology labs, what the image of that particular corporation was.
Yeah, it was the umbrella, right?
Umbrella Corporation. So who is Umbrella Corporation? It's probably one of those corporations people have probably never really heard of, and they would like to keep it that way. It's one of those that probably should be investigated. But think of how many different labs across the world where a lot of these bio weapons and bio things and whatnot take place. Right? We know that Fauci and company through the NIH and Obama 44 were in cahoots with Wuhan. But there's also labs in the eastern part of Europe where a lot of these things are done as well, over near some of the Balkan states next to Russia. And back in 2017, I think it was, I want to say Southfront had a very good article on that I archived. And the thing one single article ended up being about 100 pages by the time I finally quit archiving all the PDFs, all the frickin' videos on that were attached on just all of the stuff and that they were involved with. And of course, this is all dark money stuff, under the radar. And how many more of these things actually exist where these experiments are actually taking place? And at the end of the day, one has to ask, cui bono? who really benefits out of all this? You know, you think of people like Peter Thiel, who most people know from Palantir and everything else, but obviously he has a vested interest because he's one of those that foresees a day when humans will have infinity life. They'll just edit their way out of it. So, and in conjunction with that, think of Big Ag and Big Pharma and how they work behind the scenes to develop these things to perpetually keep us sick and on the wheel. Never healthy, by the way, but always having to go back to them for the next solution.
That's where I remember his name. It says he was Cass found himself in national spotlight when he was appointed chairman of the President's Council on Bioethics, which you just brought up, in August of 2001. It was left to Cass to nominate many of the 18 members of the council, all of whom had to be approved by President George W. Bush. There you go. So he's not only part of what you just brought up, he's also the person that chose the other members of the council, many of the other members of the council, and George W. Bush apparently had to approve everyone. That's where I remember the name. Down the rabbit hole it goes. All right, sorry, I was like, I knew I'd heard that name. I was like, how do I know this name? So, yeah. I don't know. I think that these are people who are kind of sick. Not kind of, they are sick, and what they're capable of doing and what they have been doing behind closed doors in other countries. If anything's, if they get too much attention in America, they'll just go somewhere else to do it. And that's what I think Breaking had sent, was that video that went along with that thing that we were reading earlier. But the Dr. You had brought up, what's that other article I think you had sent as well? Oh, this one, just to highlight something else he had shared. If there's any question, this was the breakpoint that he had shared, where it says in 2018, a Chinese scientist announced he had used CRISPR to genetically modify human embryos. At the time, more respectable scientists announced his actions as unethical, given how new the technology was and how little ethical oversight there was for using it. In essence, this dangerous technology has been released into the world with no limitations except play nice. So that's the only, I mean, there was no real criticisms. Why was the ethical outrage so shallow? You know, I mean, it's just that it seems like no one has got their finger on it. It's the same thing I'll say, you mentioned briefly about, if you'll remember, and I know I've said this before, about what Elon Musk, if you go back in time, what he was saying back in like 2013, 2014 about AI, and he was warning about it. You know, you're going to, using the use of AI is dangerous. No one's put any restrictions on it. And, it's like, he warned about it. And then when he did another interview here, I believe it was last year, and asked about it, and now he's coming up with all this nanotechnology. He was like, are you now not concerned? And he said, no, I figured if you can't warn people, they don't want to listen to you. If you can't beat them, kind of join them, was his philosophy at this point, I guess, about the dangers of messing with things you know little about. And you're rushed to be the first. And now we're messing with genes and DNA and mixing humans with animals. I mean, it's time to, it'd be nice if these people took a step back and really thought about what they're doing.
But it's not their nature at the end of the day. It's not their nature because they want total domination. They want total control. They want full-blown authoritarianism. And they want to have it for themselves for infinite life. And they think by doing so that they can be their own gods. Omniscient, omnipotent, omnipresent. And it's just not so.
This is it. I found it. And I'll just quickly bring it up to show you. I'll drop the link too down in the description. The biotech scientists now raising human animal chimeras on U.S. farms. Like on farms. creating a manimal behind closed doors, according to interviews with 3 research groups based in California and Minnesota. Approximately 20 pregnancies of pig human and sheep human chimeras have been produced in the last 12 months in the U.S. Thus far, there's no scientific papers that's been published. Of course not. They can't believe these people found out about it. Keep your mouth shut. The technology that makes gene editing possible involves altering the DNA of a pig or sheep embryos, which can make them genetically incapable of creating a specific tissue. Again, that says incapable of creating a specific tissue. Scientists then add stem cells from a person, which they hope will develop into the missing organ with human tissue. And this was back in 2016, January of 2016. Again, if anybody's worried about whether or not this is happening, there you go. That's in America.
So where do we go from here?
I don't know. I think it's wise to make people aware of what how science is. They're just kind of going rogue, in my mind. You think about what they're doing, but they're not going to. So what do? You know, what do you do?
Well, I think a lot of people are going to have to get hurt before anything's really done. It's very sad, but I think that's what's going to happen. I think a lot of people are going to get hurt.
Mm-hmm. And I'm going to drop this link too, by the way, in chat. This looks like a really good article. I didn't have a chance to read it, and I'll put it down below as well. I'll add it to keep a string of articles we've gone over to put below. But yeah, it's interesting stuff for sure. I don't think it gets people pay enough attention to this, and I understand why. You know, with politics, everything else going on in the world, people are really busy. Talking about those subjects. This to me is just as important because, again, no one's regulating it. It does not matter who is president with this stuff. They're continuing to do it. It doesn't matter who's in charge.
Yep.
Does anybody have anything else to add? Breaking, have anything else to add? We've been going for a while. And we can always go over more of the human chimera if you're interested. List below. We spend a lot of time with TMC, which I appreciate, because I think it's important to get through that to show you all the things they're capable of doing and manipulating. And so it sets the precursor for when we say, we bring up the other subjects. It's not so hard for you to swallow. I mean, it may be hard to swallow, but the proof is in the pudding. He's kind of laid it out there for you. And again, patents, I would agree, are a good source. Just if they're asking for a patent for it, it's not because they're just letting it lay on a shelf somewhere. To me,
I'll add to that. If it's a patent that's been put into the public sphere, then it's not laying on the shelf, because it can be patented and not necessarily published.
Yeah, and I think Homesteader had you go over that a little bit a couple of maybe shows ago about how hard it was to actually get a patent. It's not an easy thing to accomplish.
It's a long time.
Mm-hmm.
A lot of times it's 7 to 10 years.
I guess depends on who you are and how much money you have.
Knowing from working for corporations where plenty of patents and that are issued, it definitely takes a lot of time. I've been through the process.
Oh, it's not good. Well, all right. If you guys don't have anything else to add, we'll go ahead and call this a night. We've gone over a lot of stuff. Hopefully,
Enough to make your head spin.
To absorb. Yeah, have to go back, stop, think about, do your own research, look it up. If you don't believe us, say do your own research. I have myself, and again, if they're going to show me a study from the 70s where they were already doing this in the 70s, just think where they are in 2020. And so I think if you guys don't have anything else to add, we'll call it a night. I'll put the links down below to some of the stuff that we covered, because I think it's important, again, you look at this for yourself. And I appreciate all you guys being here tonight. It's another late night, and you know, these Fridays are always full of information. They're very interesting to me. So I love the discussion, the fact that we have them. So do y'all want to say good night or farewell or
Good morning.
good, exactly. Good morning.
Yeah, what's for breakfast?
Yeah.
Exactly. Oh, I got a breakfast casserole. This is, hey, we didn't bring up the kitchen much. My breakfast casserole was a success. I haven't made 1 year, so yay for me.
Yay for you.
I know, right? Okay, with that, we'll close this out. We'll be back again. We'll be doing this again. Been doing this for several Fridays. Again, we've only recorded, I think this is what, our fourth, which we broke down into a fourth and fifth, but I think we've got a pretty good flow here, and everybody always has something interesting to bring to the table.
Yeah, next time it'll be after the election.
Exactly. Hey, I thought about that too. Would there be something that we discussed earlier that happens after the election? We want to get together a little sooner, but that's completely up to you guys when you're available.
Yeah.
I know your schedules are, you know, difficult sometimes because of all the stuff that you guys are doing. So I do appreciate again that we could do this at least once a week. And with that, we'll say farewell and thanks for hanging out with us. And again, if you have any comments or questions, put them down below. And if you have anything you want to see us go over, you know, kind of what everyone does. I get into the symbology stuff. I've thought about going over the narrow gate because it's full of information. And again, I'll drop that link down below as well. And with that, we will say good night or good morning, wherever you are and whenever you're watching this. And we will see you. We'll see you next time. Say bye, guys.
Toodles.
Bye-bye.
See you next time.